All right, so this is going to be your immune system review and practice questions. Okay, so the first thing, this is the picture in the Campbell book. You see the first time you're exposed from the antigen, which is the yellow up top. The antigen or the pathogen is engulfed by the macrophage. Okay, so on your handout today, we actually we put the MHC molecule, the 2, on there. Ooh, that is bad. Let's try that again. I'm not very good with this, sorry. Okay, so there's your MHC2 molecule, and remember that's the antigen presenter, so it actually presents its little antigen right there. So macrophages present antigens, and they also stimulate helper T cells. Well, the name of the message that they stimulate with is IL-1, interleukin-1. Remember that's inter, between, leukin, white blood cells. That's the first message between white blood cells. And that stimulates to the helper T cells to release IL-2. So this is IL-2. Okay. So this is IL-2, and this is IL-2. IL-2 is released from helper T cells, and it stimulates the cytotoxic T cells and the B cells, while IL-1 was this one right here. So IL-1 comes from the macrophage, IL-2 comes from the helper T cells. Okay, so the B cells release, these are the naive B cells. They don't know how to make antibodies yet, but once they figure out how to make antibodies with the right DNA sequence, they become plasma B cells, and they make antibodies. Okay, well, an antibody is just a chain of amino acids. So here's your chain of amino acids, and it's got the heavy chains and light chains connected by the disulfide bridges. And disulfide bridges, that's just your tertiary structure, right? Well, antibodies, they connect to the antigens of pathogens that are floating in the blood. And so the pathogen's actually floating in the blood. Okay. Eventually the plasma cells become memory cells, and they don't necessarily make antibodies, but they know the sequence to make antibodies for that one pathogen all the time. T cells actually release perforin, which is a chemical that punctures the cell membrane of infected cells. So T cells act on infected cells, while B cells act on the actual pathogen. Read this down here at the bottom. This is important. Okay, so it says B cells defend against extracellular, that means outside the cell pathogens. So pathogens that are just floating in the blood, making the pathogen easier for target cells, or for easier targets. So flags them for the phagocytes. Okay, and then on the cell-mediated, the T cells, cell-mediated over here, defend against intracellular pathogens, so cells that are infected, or pathogens on the inside. So T cells release that chemical and lyse the infected cells. Okay, so I'm going to try to draw out what we drew out in class. I don't know how that'll work, but we'll try. Okay, so remember, we're all talking about the immune system, and there's the innate immune system, and then there's the acquired immune system. Okay, remember, innate, that's the immune system that you're born with. It's nonspecific. And the first line of defense is skin and mucus. And remember, those are just your barriers. And then you also have like inflammation, fever, etc. Right? Okay, well, the big guy over here on the innate is your phagocytes or macrophages. And I'm just going to write PHAGE in here for eater. So macrophages are phagocytes, and they have on their cell surface MHC2 antigen presenter. So they present the antigen with the MHC2 molecule. So the macrophage presents the antigen after it destroys the pathogen. So the macrophage will destroy the pathogen, eat it, chomp, 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 and it'll break it down with its lysosome, and then it'll show a piece of its antigen with its MHC2 molecule, and it does that for two reasons. One reason is to show other macrophages, look what I've killed, you go kill it too. And then it also does it for the helper T cells, which we're going to talk about in a second, so that the helper T cells will know the antigen that's been inside. Okay, so over on the next side, you've got the humoral, which is in the fluid. So this is when the pathogen is still in the blood. It's not in a cell. And then you've got cell-mediated. Remember I told you when that virus infects, and that says cell-mediated, when the virus comes in and infects, you have viruses that are floating in the blood, and then you have viruses that are in cells already. So your immune system's got to be able to get rid of both of those. So the humoral response is going to involve your B cells. And B cells are going to make antibodies, but the cell-mediated response, there's two different cell-mediated ones. And you've got helper T cells. Ooh, this is going to be hard. Okay, so let's just go with helper T, and then you've got cytotoxic T cells. And those are your killers. Those are your big guns. Okay, so the macrophages release a chemical message called IL-1. And IL-1 signals to helper T cells, hey, we need some help. Interleukin-1. Okay, all that is reception. Reception right there. Reception, transduction happens inside the helper T cell. And then the response from the helper T cell is going to be to release a message to the B cell and a message to the killer T cell. So reception is going to think about it. Transduction, it's going to respond, and the response from the helper T cell is going to be to make IL-2. So this orange right here is IL-2, interleukin-2. Okay, well, the B cell receives this message right here, the reception, and its response is going to be to make an antibody. The response is going to be to make an antibody. Antibodies attach to the antigens that are floating in the blood, and they inhibit it from doing its job, but it really marks it so that the macrophages can eat it. Okay, the reception here is going to be to make that toxin, perforin, so that it can damage infected cells. So this is damaging infected cells, and this is damaging the pathogens in the blood. Okay, one interesting fact we said was that people with AIDS, their helper T cell is infected, so they don't have the interleukin-2, and the B cells and cytotoxic T cells don't work correctly. Okay, so pause the video for a second, look at these words, and see if you can identify all these words and know the information about them. Okay, so pause the video again, check out this question. Pause the video between every one of these questions so that you can make sure you get the right answer, or you can try to get the right answer, and then I'll go over them. Okay, so the question is asking you about a cell-mediated response. Right off the bat, you should have known that the correct answer was not C, with those antibodies, and the correct answer was not B, with those B cells. So remember, cell-mediated is going to be T cells. This one has a macrophage and a helper T cell. I believe that's cell-to-cell communication because that's probably the MHC2 and that's probably the CD4 on the helper T cell. But the best answer for cell-mediated response is going to be the A. And that's going to be the cytotoxic T cell releasing the paraffin to kill. Okay, so check out this question. Okay, so question's talking about AIDS, a retrovirus that causes AIDS. It tells you that in the question. Which type of human cell is the virus infecting? And it's D, the helper T cell. Should have known that without even looking at the picture, but you would have known that by looking at the picture because I guess because it's releasing IL-2 somewhere, but I don't know if it says that. I can't read it really good from where I'm looking. Okay, the next question, it says 23, talking about which of the following HIV viral characteristics most impedes the progress of developing a cure for AIDS. So which of them is best for developing a cure? The correct answer is C. Mutations occur because HIV mutates so fast. They can't find a cure for it. But if you read through, you can eliminate the other ones. A, once the virus integrates itself into the host DNA, various introns are inserted, causing numerous mutations. So B says once the virus particle is made, the proteins are very unstable. So it's not the proteins, it's the RNA. It's not the DNA, it's the RNA. Because remember, I told you that AIDS is an RNA. It's a retrovirus with RNA. Which of the following best explains why the HIV infections is so devastating to the immune system? Well, it's because it destroys the helper T cells, which would make the B cells and T cells, killer T cells, not work right. So the best answer there is D. It destroys the cells used to select and clone B cells. So it's not inflammation, it's not lymph nodes, it's not stem cells, it's B cells and killer T cells. Okay, looking at for plants. Which of the following best represents the role of salicylic acid? Remember, that's what I told you. Oh, aspirin is salicylic acid, and that's, let's see, there we go. Salicylic acid. Correct answer is going to be A. It acts as a local regulator to signal initiation to the systemic acquired resistance. So should have known systemic acquired resistance there. And it just doesn't, those other things are just false. So the correct answer is A. 8, the hypersensitivity response. That's where it can cut off part of its body to prevent the pathogen from getting any further. Correct answer on 8 is C, the rapid apoptosis. Apoptosis is programmed cell death. So you may not have known that one. Okay, the following diagram represents an innate, nonspecific immune response for a particular pathogen. 16 says, once activated, which of the following responses would occur as a result of the TLR5 receptor? So you just have to look at the picture and see that. Right there, TLR receptor. So which of the following responses would occur as a result if this is innate? I think I didn't record me going over 16. Talking about on 16, it's the innate. So you had to have known that innate is inflammation. It's not antibodies, that's acquired. Cytotoxic T cell, that's acquired. helper T cell that's acquired. So the correct answer had to have been D. On 17, what makes this pathway most effective in fighting pathogens? Okay, well on this one we're still talking about innate. I don't know if you would have named this one. 17, the correct answer is B, the TLR5. It is a receptor that allows it to bind to the flagellum. And you can see that in the picture. That was kind of a hard one. But I will say the white blood cell diversity, because that was my second choice, but that's acquired. And then that would be specific, and innate is non-specific. So A says it has able to change shape depending on the pathogen. That'd be specific. And C says an antibody, which would be acquired. So hopefully you got it right. That one was B. Okay, this question. Okay, so looking at this picture, you're trying to see which order would be best for the adaptive or the specific immune response. And I knew right off the bat the helper T cell and the B cell want to connect so that the B cell makes antibodies. So A is the worst choice because this helper T cell is way at the end. It's like it's perfectly backwards. But if you look at the picture, let's before we just go off of it, the B cell and the T cell, the helper T cell connect. And then that little blue thing, that's the antigen, almost like it received the antigen, it used the antigen, and then it was able to make antibodies from it. What's wrong here with B is that it's already got that antigen, and then the helper T cell is way over here. So the correct answer is B. And I believe this is your last question. Okay, so the picture's showing you that a plant, a Drosophila, that's a fruit fly, and a mammal, they have a similar receptor Y. Well, anytime you see similarities, you want to automatically go to evolution. The worst choice, I think, is C. It says the pathogen that infects plants is similar to animals. Pathogens are specific, like the tobacco virus infects the tobacco plant. It doesn't infect people. So C is the worst choice. The correct answer is B, evolution. But let's read through A and D. The receptors are found only on leukocytes. Well, plants don't have leukocytes. Plants don't have blood. And then D says, so A is not right. D says the plants, Drosophila, and mammals all contain roaming phagocytes. Again, plants don't have phagocytes. So I hope that helps.